we have about we have 15 students in the class. I think one or two is missing. So, okay, we will continue from the last slide. We talk about the transport of protein um, through the ER membrane. So in the last lesson, we already discussed about how the protein is sorting and transported to different places in the cytoplasm. So there are three fundamental transport pathways for the protein. They are vector transport, membrane transport, and vesicular transport. So today we will go to the third pathway of protein transport inside the cell. So last time we stopped at this slide, I think at this movie showing the transportation of the protein, of the stool protein and the membrane protein into the ER. So after this protein come to the ER, and most of the protein are conventionally modified in the ER organelles. Many proteins are glycosylated on asparagine. This is one amino acid stay on the polypeptide chains when the protein come to the ERs. So whenever the polypeptide chain express the asparagines in the sequence, so this at this site the protein will be glycosylated. Um, the glycosylated was is taken by the addition of branches oligosaccharide side chains, and each oligosaccharide side chain is transferred as an intact unit to the asparagine amino acid from a lipid molecule called dolichons. So you can imagine that the dolichons will attach to a branch oligosaccharide group ready on the cell membrane, on the ER membrane. So when the inappropriate asparagine in the growing polypeptide chains enter the ER lumens, so the dolichon will transfer the branch oligosaccharide to the asparagine amino acid on the growing polypeptide chains. And this action is catalyzed by the enzyme named the oligosaccharin transferase. Oligosaccharin transferase. So asparagine that are glycosylated are always present in the three peptide sequences as asparagine and another amino acid, and then serine amino acid, or asparagines, another amino acid, and threonine. So that is a, the sequence of the amino acid fragment that the asparagines will be glycosylated whenever the polypeptide chain enters the ER organelle. So the X that means the X amino that means almost amino acids. For example, we will have like asparagines and like glutamic acids, and then serine, or asparagines, valine, and threonine. So whenever the asparagine uh, close to serine and threonine by one amino acid, so this asparagine amino acid will be glycosylated on the polypeptide chains. So that is one type of the modification of the protein whenever they come into the ER. And then the protein either can be stay on the ER lumen if they contain specific specific sequences on the C terminal, or this protein will be transported to the other organelle, like Golgi apparatus. So for the next transport, if the protein need to transport it to the other organelle, so they need the third pathway of protein transport called the vesicular transportation. Also, the protein, when they enter the element, they need to be folded in the correct conformation by the support from one type of the protein named the chaperone. So chaperone is one family protein that help the protein to fold in the correct form or correct transformation or correct structure, 3D structure. And if the protein is mistake in the folding, so the chaperone also help to repair the folding of the protein. And if the protein is cannot, okay, I take the message for food, okay. And in case the protein cannot be repaired, so this protein will be sent out of the ER to the cytoplasm, and in the cytoplasm, this protein will be under the ubiquitination. So this is one process that gives the signal to the protein to be sent to the proteasome, one type of the organelle, to degrade the protein. So this process will ensure that there is no mistake or defect protein appear on the cell, because the defect protein can give some harm to the cell. For example, they can play the incorrect function, or they can interact with the other protein to make to transfer the mistake to the other protein. So this protein will be degraded inside the cell if they cannot fold in the correct form. So this is another type of protein modification on the ER. So you can see that in the ER lumens, or in the mitochondrial matrix, or in the space between the two membranes of the mitochondria, so always stay the chaperone protein to help to fold the other protein in the correct form, and also inhibits the incorrect interaction of the protein to the other molecules surrounding them in this space. Okay, so the protein need to be transported to the other organelle, like Golgi apparatus. So they need to use the third way of protein transport inside the cell. We call the vesicular transport. So entry into the ER lumen or membrane is usually only the first step on the pathway to another destination of the protein and the other molecule, like lipid. And for the ER lumens, that destination initially, at least, is generally the Golgi apparatus. So the protein and the lipids are modified in the ER and then sorted. Oh my God. I'm sorry, the internet is a little weak today, so we just out. I will try again. I will show again the. Can you see the slide? Yes, I think. Okay.
I don't know if you have bad
Sorry.
connection or not, but I doesn't seem. I don't
So the
think.
protein after enters the ER and modified and sorted, and it will be transferred to the Golgi apparatus. This process is carried out by continued budding and fusion of transport vesicle. And this vesicular transport extends outward from the ER to the plasma membrane, where it allows protein and other molecules to be secreted by a process called the exocytosis, and it reaches the inward from the plasma membrane to the lysosome, allowing the extracellular molecule to be imported by the process called endocytosis. So this is endocytosis, and this will be the exocytosis in the cell, involving in the vesicular transport. So together, this pathway that provides a communication between the interior of the cell and its surrounding. So involving in the vesicular transport, we also it can be sorted or divided into three pathways, like the retrieval pathway, endocytotic pathway, and biosynthetic secretory pathway. So for the endocytotic pathway and biosynthetic secretory pathway, uh, I already talked to you. For the third pathway, the retrieval pathway, that means the cell try to reuse some material from this organelle to the other organelle. For example, whenever the vesicle is transported from the ER to the Golgi apparatus, so some type of the protein on the ER membrane will be transported to the Golgi apparatus, and the cell also recycle to use this protein by transport back this protein in another vesicle back uh, to the ER. So by this way, the cell reuse transmembrane protein and the other material in the vesicular uh, structure. So we call this the retrieval pathway. So you can see this retrieval pathway can be transported both from the plasma membrane to the other organelle, like the endosome, lysosome, Golgi apparatus, and back to the endoplasmic reticulum, the ER. So that is a three pathway involving into the vesicular transport. So even they have three pathway, they all together have the similar step during one vesicular transportation. So every vesicular transportation will contain this four steps as indicated in the slide. So here you see from the donor compartment to the acceptor compartment, the cargo means the cargo here is a protein. So we have two types of the cargo. The TM cargo, that means the transmembrane protein, and the solute cargo, that means the solute protein. So for this transport, we have two types of cargo, the transmembrane protein and the solute protein. So this protein now present in the donor compartment, and they need to be transported to the acceptor compartment. To do this, the cell first need to form one structure called the vesicle. So the first step is the vesicle formation. This the vesicle formation involved in many types of the protein, like the coating protein and the protein with and this action also needs energy from the hydrolysis of GTP molecule, and also they need the adapter protein to make the interface for the co-protein bind to the membrane at this site. So the cargo will be collected or focused on this site, and after the protein is collected, the membrane at this site will be how to say blend, or they make the budding in this size. So the next step from the vesicle formation is budding. You see the membrane at this side they develop and bend and make vesicle form, like you see here. After budding step, the protein, the cell will move to the another step. I'm sorry for a moment. Okay, so the third step in the vesicular transport is the vesicular translocation. So you can imagine, like after the cargo um, is uh, is put on the vesicle, and this vesicle will be transported through the cytoplasm from the donor compartment to the acceptor compartment. Just like in our life, that's like the when you put the cargo on the trucks, and the trucks will carry the cargo to the other place. So as a third step, the vesicle will be translocated by the cytoskeleton and the motor protein. So vesicle will be bind to the motor protein, and the motor protein will work on the cytoskeleton to the acceptor compartment. So in the cytoskeleton lesson, we will understand more about this step. So today I will quiz this step. You just remember that the vesicle will be translocated by the motor protein on the cytoskeleton to the acceptor compartment. So, and after they go close to the acceptor compartment, so they will train to the next part we call the vesicle tethering. So it's like um, when the task or when the boss they go to the to their target place, and they need to do the tethering. I don't know if you understand the word tethering. Like on the boat, they will put something very heavy to the to the um, to the water, and by this way they will stop the boat near to the um, near to the port. Okay, so it's also similar to the vesicle. That the vesicle chattering is start it started when the vesicle is close to the acceptor compartment. And also this process, the vesicle chattering also needs support from many types of protein on the vesicle coat, on the vesicle membrane, and on the acceptor membrane. And after the chattering is finished, so the fourth step will be taken place, and the fourth step is vesicle docking fusion. That means the vesicular membrane will be fused with the membrane of the acceptor compartment, and during the fusion, all the cargo protein will be pushed into the acceptor compartment or located on the membrane of the acceptor compartment. So that is the fourth step of vesicular transport, and you can see many types of the protein involved in this process. Like here you see they explain about each type of the protein involved in each step of the vesicular transport. So for the vesicle formation, you need, of course, first the cargo protein like transmembrane cargo or sloop cargo, and coat protein like creatine, COP1, COP2, and the small GTPase like REP, RF, or the adapter protein. For the budding protein, you need the support from the dynamine protein, and for the vesicle translocation, you need support from the motor protein. For the vesicle tethering, you need support from the vesicle tether protein on the acceptor compartment membrane, and also the other tethering protein like COG or EN also needs energy from the GTP hydrolysis. And for the docking and fusion, you need support from the snare protein. We have two types, the V snare and the T snare, and also the trans snare complex when the fusion happens. So in the next slides, we can go in detail of the step for this type of transport. Okay, so for the first step, also very important step, it is a vesicular formation. Um, so this slide is spread about the first step of the vesicular formation from the creatine protein, the the coating protein named creatine. So creatine coated vesicle transport first the we select the cargo molecule. In this part, in this slide, because the vesicle is formed from the creatine, and creatine they located on the cell membrane, on the plasma membrane. So this, in this line you see this is the extracellular space, and here is a cytosol. The creatine will make the vesicular on the plasma membrane. So in the first step here, the cargo receptor, the blue one, you see, the cargo receptor will bound the target protein, and after the binding of the cargo protein, the cargo molecule expressed as a red dot here, bind to the receptor. So this binding will be induce the capturing of the receptor by the adaptin protein. So this complex, the cargo and the receptor, will be captured by the adaptin protein. And this adaptin, when they come to bind to the receptor here, they also bind to the creatine. So this clathrin molecule will be bind to the cytosolic surface of the membrane at this site. And when the more clathrin come to bind to this side of the plasma membrane, they induce the budding of the membrane at this site. So that means the membrane will be blended at this side and developed inward to the cytosomes. And then they make the structure like vesicle, as you can see at the next step here. So when the budding is finished, another protein named dynamine protein. So this is also one GTP hydrolyzed protein. So dynamine will bind at this site between the vesicle, the budding, and the plasma membrane. So dynamine will assemble surrounding the neck of the budding vesicle. And once assembled, the dynamine molecule will hydrolyze their bound GTP to produce energy and help the other protein recruited to the neck to make a complex. And this complex will cut the link between the budding and the plasma membrane and pinch up the vesicle from the plasma membrane. So after this step, the one complete vesicle is formed with the clathrin coat surrounding the membrane, and this clathrin coat can interact with the membrane of the vesicle through the adaptin protein. So adaptin protein somehow they play like interface for the coating protein form, for the coating formation surrounding the vesicle. So after budding is complete, the vesicle is transported to the target or the acceptor compartment, as I mentioned before. And whenever they close to the acceptor compartment, so the coating is how to say, the coat protein are removed. So the naked vesicle now can fuse with the target membrane. So to do the fusion, just remember to do the fusion, the coat must be removed before the vesicle fuse with the target membrane. So you see seven types of protein involving in the step, like the adaptin protein, support for the coating formation, and the coat protein like clathrin to form the vesicle. And the dynamine protein help to pinch up the vesicle from the donor membrane. And after that, the coat protein need to be removed to have the vesicle do the fusion with the target membrane. So in the first step, you see many types of protein need to be present to form the vesicle. So clathrin molecule form the basket-like case that have shape membranes into vesicle, and dynamine assemble so that the neck of budding vesicle help to pinch up the vesicle from the plasma membrane. Okay, so I have one question for you. Sorry, I will give some on the. Sorry, I forgot to put it in the slide, but I will release it on the um, yes, on the class in the classroom, so you can thus help you to understand more about the function of each type of the protein involving in the vesicle transport. So the question is, um, which of the following is a function of adapter protein? So I would like to ask about the adapter protein. I want you to remind about what we just learned. You can use the slide as a handout I sent to you and see the function of the adapter protein. So in the slide, the adapter proteins named the adaptin. I don't know if you already received the question. Have you received the question? Have you received the question?
Yes.
Because in my computer they still um, give the name like asking. So I don't know if you already received the question or not.
We got a question.
Okay, thank you. So the internet from my computer is very bad. I will turn off the camera with hoping that it will increase the internet speed. I hope. Let me see. Oh, oh, it's too bad. Now I cannot join to the classroom.
I still see you on the meeting.
Yes, yeah, thank you. Yeah, I'm still on the meeting, but I cannot come to the classroom to see if you already give your answer or not. So that is the problem. Cannot see. Um, all of you already return your answer or not on the classroom? Maybe not. Okay, I'm sorry for the internet. Okay, since I cannot join to the classroom, so I don't know if you already give the answer or not. But we will discuss about the question, and so the question is, which of the following is a function of adapter proteins? So can you give your answer directly to me now? For example, okay, I will call some of you, maybe one or two. Okay, I see. Okay, Ming, what is your choice?
I actually still haven't fully understand everything, so that's why I choose the answer A.
Answer A. Can you speak out loud of the answer A? What is the answer? What is the answer A? I cannot come to the classroom, so I cannot read again about the question. Okay, yeah, you can type on the chat window. And what okay, you pick randomly. So Thảo, Vũ Bích Thảo, what is your answer?
Um, at the first time I chose B,
Yeah.
but now I'm a little bit confused with C. With the C. The answer B is they play a role in vesicular targeting, and C is they attach tethering protein.
Oh,
I'm a little bit confused about two, two
you
choices.
try this.
Yeah.
Okay. Um. So okay. Uh. Just look at this. What is at this slide? So you see, here the adapter protein in this case is adaptin. Okay. So adaptin is one type adapter protein. You will see in the next few slides. You will see that each type of the vesicular they need support from different type of the adapter protein. But in this case, the adapter protein is adaptin. So adaptin will support for the formation of the vesicle by play function as the interface for the cargo for the coating protein come and bind and form the vesicle. So also, you see the adaptin, they only bind to the receptor whenever the receptor they recognize and bind to their cargo, to their specific cargo. So in somehow the adaptin also help to select the suitable cargo for the transportation in the vesicular form. So among the four choices I give to you, so the most suitable and the most correct answer for the function of the adapter protein is they serve as interface for coat formation. So that is the right answer for this question. Okay, so uh, I don't know if any of you select or choose this answer, but that is the correct one. So there are many types of the seven types of the coating protein, and like you see here, now the scientists determine three types of the coating protein or coat protein. They are clathrin, COP1, and COP2. And these coat protein, they locate in the different in the different location in the cell. For clustering, as I just give you in the previous slide, so clustering protein mostly they locate on the plasma membrane or near the plasma membrane to help form the vesicle from the plasma membrane during the endocytosis. For the COP1 protein, the COP1 protein mostly locates on the Golgi apparatus, and they will help to make the vesicle from Golgi apparatus to transport to the other parts inside the cell. For example, come to the back to the ER or come to the lysosome and endosome. So COP1 located mostly on the Golgi apparatus, and COP2 located mostly on the ER membrane to form the vesicle from the ER and transport to the Golgi apparatus. So that is a three type of the COP protein: creatine, COP1, and COP2. And each COP protein needs support from different type of the adapter protein. For creatine, they are adaptin, and for COP1 and COP2, they will be the SCF or SRF or ARF. In the next, I will give you some example in the next slides for this formation of the vesicle from these two types of COP protein. So here in the table, explain about some types of the code vesicle, and you see that was a cluttering code. So they need the creatine with the adapter like adaptin protein, and they can from the Golgi apparatus to the lysosome and then to the cell membrane, plasma membrane, and also from the plasma membrane to the endosome, or the other coprotein like COP protein, COT. So the COP protein needs the other support from the other adapter protein, and they can be from the ER or Golgi apparatus and exchange between these two organelles in the cytoplasm. Okay, so we have a short break and come back at 10 a.m.